certain numbers that are following certain letters, suffix that follows the allele, like sometimes you find an N or L, that mean that the expression, it relates to the expression of this antigen. Like if it's a null allele that's not expressed, or it has a low expression, and this plays an important part when we are detecting those antibodies and determining if they have, if they react against the patient or not. The HLA typing, the gold standard for HLA typing since the late 1960s were the complement-dependent cytotoxicity, or the CDC. And this was first described by Dr. Terazaki and in the 1960s. And as you can see, we use a Terazaki plate. Each well of this plate has a separate anti-HLA antibody that we obtain from many sources, like monoclonal antibody or multi-virus woman antibodies, monoclonal antibody that we manufacture in the lab. And when we incubate the target cell, the patient cell, with these antibodies, if it carries the same HLA, they will bond together, and when we add complement, that will lead to activation and eventually to cell lysis. This cell, and that's if we have the antibody and we form the antigen antibody complex. If not, there will not be any cell lysing. And this cell lysing can be detected by many methods, depending on the staining mainly of the cell, and one of which, like you can determine, like if this cell is carrying, this is the positive where the dye penetrated the cells, and this is negative where the dye did not penetrate the cell because there wasn't any cell lysing. And we read multiple wells enable to us to determine the HLA typing. So for example, if this cell is carrying A2, B7, and DR4, that will indicate that this patient has the HLA typing of A2, B7, DR4. But we have a very stringent criteria to call this antibody when we are using complement-dependent cytotoxicity. We have to have multiple positive wills in order to be able to call that. So the standard called for 3 separate positive reaction in order for us to be able to call this antigen. So, which will make it very difficult because we will need a lot of cells. The obstacle that we were facing with cytotoxic dependent cytotoxicity in HLA typing was the cell quantity. We need a good amount of cell to enable us to test for the diversity of the antigen. Also, the cells need to be viable, because the true measurement of the positive and negative are the cell viability. So the cells need to be viable, and this will mean that we cannot receive any specimen that is more than 2 or 3 days old. And of course, it's only an antigen expression or serotyping. It's not allele expression or intermediate, low resolution, high resolution typing, and very limited specimen source. Not every specimen that we can use for cytotoxic antibody secrene, complement-dependent cytotoxicity for HLA typing, because there are certain interference factors in some of the anticoagulants that will not allow us to be able to carry this procedure. And since then, in the 1990s, since the beginning of the 1990s, we start to develop some newer molecular-based HLA typing techniques, which will increase the accuracy and reproducibility of the HLA typing, and also there are significant advancement of the molecular techniques that to provide you with the allele level resolution and maybe the higher resolution. Also, the unnucleated cells will be a good source of DNA that can lead to the detection of the HLA typing by molecular, and even like any tissue, any blood tubes, if it's whole blood, can be used to extract DNA, and so to perform the HLA typing. And it's open for automation. The molecular can be automated easier than the CDC, which is, as you saw, is a labor-intensive manual type of testing that cannot be automated, and And so the time factor and the higher throughput is an advantage with the molecular HLA typing. The major molecular HLA typing in use are the SSB, which is sequence-specific primer, and SSOP, sequence-specific oligonucleotide, and the SPT, sequence-based typing. And there are other like flow cytometry-based or real-time PCR, but mostly used in research, not in a clinical lab. So most of the clinical lab, you will see SSB, SSOB, and SPT as the technique in use for the molecular HLA typing. And of course, the HLA typing, molecular HLA typing, we are shifting from the serotype to the allele level type. Like A2 by CDC will be HLA A O2, asterisk O2, to indicate that this is an allele and it's a molecular HLA typing. The same with B7, C8, and DR4, DQ3. The first technique that I'm going to talk about is the SSP, which is the sequence-specific priming technique that we started using in the mid-1990s, 1995, 96, and it has a very simple but complicated steps where, first of all, we have to have a good amount of DNA as a target DNA from the patient or the donor, and then we extract the DNA, and there are many methods to extract DNA, even now mostly automated methodology to extract DNA. This DNA is subjected to very high heat in a thermocycler to denature this DNA and produce, instead of double-strand DNA, to produce two single-strand DNA, followed by the annealing part where the DNA will be mixed with a primer, the target primer. So if we are detecting, let's say, A2, this primer will be for A2 and it will anneal or stick to the denatured DNA, and followed by amplification, where using building blocks, nucleotides, we will start to build a new DNA, and will end up to amplify or multiply the DNA many times, so we end up with millions and millions of copies. That is much easier to detect than the single or the few copies that we started with. And there are many ways to detect this copies or magnified, amplified copies. Is, for example, for gel electrophoresis, where we add a dye to the amplified DNA, and then do the gel electrophoresis, and whenever this is an internal positive control, and this is a test where the internal positive control is working, and this is a positive reaction, and for example, this is negative reaction, this is another positive reaction, and knowing the different location of these primers, we can tell which one is positive, and so what's the HLA typing. For example, this is a DR low, and this is, for example, DR12 and DQ5. And the advantage of SSB that it's a very fast technique that you can get the HLA typing within few hours, less than 5 hours, if you have a good DNA. And it's really excellent for, especially if you have virtual cross-match where you need the result of the HLA typing in a timely manner. So it's excellent for this purpose. The problem is initially it will give you a low to intermediate resolution, and you need to follow this up with a higher resolution that will take more time. The second technique that I'm going to talk about is the SSOB, which is the sequence-specific oligonucleotide, which all started with when they introduced the microsphere, 100 different beads that you can coat with the probe and then detect the HLA. So this is the reaction itself, which is SSOP, which is a very simple reaction where you can extract the DNA, amplify the DNA with the target, and then after amplification, we can detect that it did amplify, and then, which is like a quality control measure, we detect the amplification, and then we couple the amplified product or the amplified DNA with the beads, and then we detect the positive reaction based on the fact that the positive reaction will be coupled to the fluorescent dye, and we can read it on the using the laser. So first, the beads are, this is the bead, and the surface of the bead are covered or coupled with the target or oligonucleotide in this case. And when we add DNA, it will attach to that particular, if present, the DNA will attach to the amplified DNA to that particular target. And then we add the second antibody with the fluorescence. And after washing, if a positive reaction remains, it will be detected when we pass it through the machine, acquire it, and it will be subjected to a laser, and this laser will emit the light on the fluorescent, and this will be translated into a certain HLA typing depending on the strength of the signal. So the analysis software does part of the work to analyze which are positive and which are negative, and so to give us the different antigen like A1, A31, B35, B40, and so on and so forth. The last technology are sequence-based typing, which are mainly used for highly polymorphic HLA class 1 loci like A and B. And what we do in the sequence-based typing, where we purify the DNA and add it to a mixture of different nucleotide. After denaturation, if positive, we'll have a group of fluorescent labeled amplified DNA fragment that we use electrophoresis to differentiate, and will end up with different sequences that we can define at a higher resolution, the HLA typing. And it's mainly used in HLA class 1, especially A and B, A, B, and C, to determine the higher resolution, as the sequence-specific oligonucleotide and sequence-specific priming is not always successful in providing you with a higher resolution in this case because of the high polymorphic region in the HLA class 1. So I'll give you a few examples. Like at the end, what do we get? We get certain HLA typing. For example, this is a patient with a certain HLA typing, and this is only A, B, and DR. But of course, for HLA, for solid organ transplant, we also do the HLA, the comprehensive HLA typing, as mentioned by Dr. Al-Mishari. We started actually since 2009 to provide the full HLA typing A, B, C, DR, DQ, and class 2 high resolution. But this is a classical A, B, DR, where the patient has a typing of A2, for example, A3, B27, B58, and this is the donor typing. As you can see, there is mismatching in A, B, and DR, and this is like a haplotype. So there is 3 out of 6 mismatches in this case. The same patient with another donor, you can see the mismatching, for example, in DR16, which is in class 2. And so this is 1 out of 6 mismatches. And for the same patient, if we have another donor, which is entirely different than this, so we can say this has a higher number of mismatches, so 6 out of 6 mismatches. So in brief, the DNA-based or molecular typing method provide an increased accuracy in the identification of HLA antigen and allele. It has some limitation, which is because of the growing number of alleles. We need always to consider like a higher resolution type of typing, especially in the hematopoietic stem cell transplantation, where we need a higher resolution. And this is where we can use the sequence-specific SPT technology, sequence-based typing, and to be able to provide you with the higher resolution typing. Thank you. I think we're going to pray.
Thank you very much, Maha. Excellent presentation. Now, because we started late, I think we'll go for a coffee break right now for like 15 minutes, if you don't mind. We'll reconvene at 10:30. for the second part of the morning session. Thank you very much. I think Fadi is here, so we can start. Very good. Okay. Good morning again, and then we'll start the second. In fact, we were told by the organizers to be punctual, so we can finish on time, and we'll have ample time for questions. The second part of the morning session will be presented by Dr. Muhib Alawami and Fadi Zayer. They will address the remaining part of the triangle of the HLA system. Dr. Muhib will start by presenting the HLA antibody, followed by Fadi Zayer, and eventually we'll have close to 30 minutes for discussion for the whole speakers. Muhib.
Bismillah ar-Rahman ar-Rahim. Good morning, everyone. Today it will be my presentation now will be a continuation for what Dr. Khalid did talk about, and also Maha. So I'll be talking about HLA antibody detection methods. Basically, in the lab, we are doing 3 key tests. HLA typing, which Maha talked about earlier, and also HLA antibody testing, which I will be talking about. And the third key test we do, cross-matching, which will be the job of Fadi Zayer, inshallah, after this afternoon. HLA antibody testing, or what we refer to as panel reactive antibodies, PRA for short. Sometimes we use it exchangeably with antibody testing, which is a screening process to detect antibodies in recipient serum that react with HLA antigens. So basically testing the patient's serum whether he has antibodies or not. If the screening process shows positive results, we say that this patient is PRA positive, as simple as that. And also we refer to that this patient is sensitized, and the definition of sensitization is the patient who have developed anti-HLA class 1 and or anti-HLA class 2 antibodies. The importance of sensitization, as my colleagues earlier mentioned, started in 1960s by Kissner and Nilsson in 1966, where he published that hyperacute rejection is associated with pre-existing anti-HLA antibodies directed against donor cells. And a few years later, Batil and Terasaki also documented that hyperacute rejection is associated with positive cross-match in kidney transplantation. Since we are a laboratorian, we are following our accreditation agency, which is ASHI, which mandate that laboratories performing testing for renal transplantation, they should prospectively type donor and transplant candidates for HLA antigens, which Maha talked about. Also, they should screen transplant candidate for the presence of the anti-HLA antibodies, which I will be talking about, and also mandate performing cross-match prospectively. This will be a repetition of what has been mentioned before, but I have to just elaborate a little bit. History, where the antibodies, how come patient becomes sensitized? There is more than one source that he can become sensitized. If he is a sick patient and he needed blood transfusion or any blood products, even if it is just backed red blood cells, still there will be some white blood cells remaining in that product, and when the patient see it, he or she will develop an antibodies against the HLA antigens on the white blood cells. And with the repetition of the blood transfusion, that means the immune system will see it again and will develop a stronger immune response against HLA antigens. Also, another source of sensitization, which is multiple pregnancies. In the first pregnancy, when during gestation, half of the genes in the baby is foreign to the mother, coming from the father. So the first time the immune system will see it, and with frequent or subsequent pregnancies, the immune system will see it again and will develop a stronger immune response. And this mothers will become sensitized for the antigens coming from the father. Also, a source of sensitization to HLA antigen is previous transplantation. Someone who has been transplanted before and he lost his graft for whatever reason through antibody-mediated rejection. That means he has antibodies against the HLA antigen expressed on the organ. Also, if a patient being transplanted and he's not doing very well, which means that evidence of graft rejection, that rejection could be a result of antibody-mediated rejection, which means that he is now becoming sensitized, even if he hasn't been before. Also, major surgery, even if it is not related to do with transplantation, just the cutting open the patient, that means he has gone through a trauma, and this will lead some B cells and other cells to produce cytokines, and what they call it cytokine storm, which will make B cells produce antibody, not specifically. Those antibodies could be anti-HLA antigens. Also infections, some of the microbes like ABV, or viruses, they share some epitopes with the HLA molecule. So when the immune system exposed to these microbes might think that this is when the immune system exposes this microbe which share same epitopes as HLA, will develop HLA antibodies. But later, when the immune system see a real HLA molecule, will think this is something foreign and will fight it. Also, vaccination. Even if someone he is a healthy person, he hasn't been transfused before, and he is becoming a donor for a renal patient, and when we test, or if you get a healthy person and you test his serum and he's never been transfused, whether patient or not, you might ask from where he gets his anti-HLA antibodies. Some of the vaccine, again, they share some epitopes with HLA molecules, and they will become sensitized to HLA, although they haven't seen the HLA molecule before, but due to mimicry between them, he or she becomes sensitized. Also, in the last few years, they were talking about natural HLA antibodies. If you test the serum of healthy untransfused patient who has never been transfused and you test his serum, you might find some of the HLA antibodies. Again, this could be due to different reason other than seeing the HLA molecule itself. There is more than one method for detecting anti-HLA antibodies. Some of them antigen non-specific, like complement-dependent cytotoxicity, or flow cytometry using cells, or some of the tests that are antigen specific, like ELISA, flow cytometry using beads, or multiplex beads, which mainly the Luminex technology. And each technique has its own advantage or disadvantage. The one Maha and Dr. Mishari mentioned was the CDC, and there is various variation of the basic CDC, or sometimes they call it standard CDC, or the NIH, which is National Institute of Health. This test can be, the sensitivity of this test can be improved by adding washing step or extending the incubation, or by adding some other agent like anti-human globulin to improve the sensitivity of the test. Again, each test has its advantage and disadvantage. There are too many. It's beyond the scope of this 30-minute presentation, so I will focus on the coming few minutes on the complement-dependent cytotoxicity and the Luminex technology, which is the main two techniques used by most of the labs routinely. The basic principle of the complement-dependent cytotoxicity, CDC for short, if we have a patient that he's sensitized, which means he has anti-HLA antibodies, and we incubate his serum with lymphocytes, again carrying HLA molecules, and we incubate them for some time, these antibodies will find their target and they will bind to it. And after this initial incubation, we can add complement, and the complement will complement the antibody function by making holes on the membrane. These holes will allow a vital dye or any stain we use to enter the cell and stain it. And this is what we refer to as positive reaction. But in case of non-sensitized patient, So in his serum, there is no anti-HLA antibodies. Again, when we incubate his serum with cells, lymphocytes carrying HLA molecules, there will be no binding because no antibody existing. So when we go through the rest of the procedure, which is adding complement, then adding the stain will not enter the cell because the cell membrane is intact. The cell is not attacked by the antibodies. And we refer to this as negative reaction. We can add extended incubation time between antigen antibody. This is a method to increase the sensitivity. We can anti-human globulin here to increase the specificity. All these techniques is possible. So I will go through this procedure maybe in more detail because it's very important. The panel reactive antibody, what we refer to as PRA, the way we do it, we extract lymphocytes from, like when we do it in our lab, we stopped using this, but it's very important to understand. We invite 60 or 70 people in our hospital and we draw blood from them. We extract their lymphocytes. It's very easy, it's not complicated. We just take their lymphocytes and we clean them and we dispense in each person lymphocyte at a certain concentration in one of these, what we call it Terasaki plate. In each well of these wells, this is 60 wells, we add a cells from one donor. And also, when we take the lymphocyte from the donor, we do HLA typing for each donor. So we know the HLA molecules on the surface of each of the lymphocyte we used. When we make hundreds of trays, we freeze them in liquid nitrogen, and whenever we have a patient we need to test, we take his serum, patient serum, and we dispense 1 microliter in each of these wells. And again, each well is with non-HLA phenotype. We know the HLA typing. When we dispense the serum to all of the wells, we incubate, then we do a washing step, then we add complement, and again we incubate for 1 hour. After the incubation of 1 hour, we add vital stain. Again, as I mentioned earlier, if the cells being attacked by the antibody, that means there is an antibody in the patient, and what we need to know is just to visualize it. So after finishing the procedure, this is how they will look like under a microscope. If they are intact cells, they will not get the stain, so they will stay refractile, and this is how they look like under the microscope. If there is antibody specific for those antigen on the cells, those cells will be attacked by membrane attack complex, and there will be holes in the membrane, cell membrane, and the stain will go inside the cells and stain it red. And you could use different stains, and it still will enable you to visualize positive from negative. So the purpose of panel reactive antibodies is really to tell the attending physician or clinician the sensitization status of the patient. Is he sensitized or not? Does he have anti-HLA antibody or not? And also by doing PRA, you will tell the likelihood of transplant negative. If he has no antibodies, that more likely the cross-match will be negative. If he has antibodies, the more he has, the less likely cross-match between this patient with any random donor in the population will be positive. Also, if the patient is not really very highly sensitized, you might tell the antibody specificity of the patient's serum. This is an example of unsensitized patient. When we added the patient serum to the donor lymphocytes, none of the wells reacted except the positive control. Always in the laboratory we have a positive control and negative control. And when you read it under the microscope, you will find just the control is positive. And we refer to the PRA as 0 PRA. And the antibody specificity, of course, since everything negative is non-detected. So this patient is non-sensitized. So if someone asks you what is the probability of this patient becoming a negative cross-match, the probability is very high, close to 100, his chance of a negative cross-match. If we take another example of someone who is sensitized, and we do the same procedure exactly, and we read it under a microscope, we score according to certain criteria by the accreditation agency. And in this example, we find 7 wells that are positive. And to calculate the PRA, we divide 7 by 58. We have here 60 wells minus 2 we used for control. So 7 over 58 times 100. The PRA of this patient is 12%. And the way to know specificity, especially if someone is not really highly sensitized as this patient, we look at the positive wells and we check what common antigens are there between all the 7 wells. And in this case, whenever we have A2 antigen on the cells, the antibodies from the patient is reacting. So we can tell with confidence, following certain criteria, that this patient has an antibody anti-HLA A2. So again, if the physician, what's the likelihood of this patient becoming a negative cross-match? So 100 minus 12, his chance is 88% becoming a negative cross-match. If he is for cadaver, his chance from his local population, his chance is 88% to be cross-match negative. But if we have a highly sensitized patient, as this example, where almost all of the wells are reacting, it's very challenging, but calculating PRA is still possible. Again, we have 49 wells positive over 58. The PRA is 84%. And the challenging part, can we determine antibody specificity? In this case, all what we can mention that this patient is sensitized and the specificity is multispecific or broadly sensitized. Because most of the well is reacting, we don't have something to rule out or to rule in as an antibody present or absent. And the probability of negative cross-match of this highly sensitized patient is low. It is 16% only. So the complement-dependent cytotoxicity, CDC, whether it's standard or AHG, it detects HLA antibodies that are capable of binding and activating complement, like IgG3-1 and IgM. If they are not complement fixing, disregarding whether it is clinically relevant or not, you cannot detect it. Also, it will detect non-HLA antibodies and autoantibodies. Challenging with the CDC, it is very time consuming and very subjective. If someone read under the microscope, especially if it is not really strong positive, if it is weak positive, you might have some disagreement between 2 expert technologists. Also, it needs a suitable panel of lymphocytes. When we ask 60 or 70 people from our institution to donate, we are not really controlling the composite of this, what we call a Terasaki tray. So we just say thank you whoever willing to donate. We don't have control of the composition. So it's really hard to find very comprehensive composition of lymphocyte unless you are very lucky. Also, it is of very limited sensitivity and specificity, especially comparing to the new techniques, as I will be talking shortly. Again, can vary widely by the panel tested. If I do PRA using this technique I just talked about today, and we run out of those particular tray, and we make a new batch, say next week, you might see the PRA is changing. Because of not the patient PRA changing, because of the composite of the panel we used. So PRA percenting can be misleading. High PRA patient does not assure positive cross-match, again due to just the composite of the panel. And the vice versa is also true. Very low PRA does not assure negative cross-match. Maybe the patient has an antibodies, but the antigen, they were absent on the panel we used to test them. But when we do the real cross-match, they will show up. Change in the antibody specificity may occur without change in PRA. Again, all depends on the composite of the panel. So it's very dependent on the panel being used. Oops. So this is what we use for many years for antibody testing. We stopped using this method and we went with the flow using the latest technology, which is the solid phase immunoassay, which has a lot of advantages over this old method. But it's very important to know, I guess, how it started, the PRA. Now I will talk about solid phase assay for detecting antibodies. As Maha mentioned earlier, the Luminex technology depends on using 100 beads, different beads. The difference between each of these 100 beads, you have 10 here and 10 here, is the color of each beads. No two beads having the same color intensity. Each bead is really different. So if we take one bead of those 100 and we label it or attach to it an HLA antigen, then we incubate the patient serum, who we know sensitized, he has anti-HLA antibody. This antibody will bind to the HLA molecule. And we add secondary antibody that is labeled with fluorescent or fluorochrome, and we can visualize the positivity using the Luminex machine, which is a flow-based machine, very small, tiny machine, but it's very powerful. So one of our patients, he has these 7 wells positive, and as these beads, which is in a suspension, very small beads, if you look at the fluid, you will see just a solution. When these beads go through the Luminex machine, which is a flow-based machine, any of these beads, for example, if this bead, which is positive, pass through the machine will know which bead it is because of the color, and it will know that it is positive because of the secondary antibody tagged with fluorochrome, and also for the rest of the positive wells. So as simple as that, you have a bead. The bead is coated with HLA molecules. If you have an antibody, the antibody will bind to the molecule, HLA molecule. Then you will do washing and incubation. Then you will add your secondary antibody. If it is positive, it's easy to detect it. The machine will do this job for you. So for example, this patient, let me go back. So this patient now, for bead number 7, for example, as it passed through the machine, it will know by laser 1 which bead it was, and also it is coated with what? HLA molecule. And the other laser is for the secondary antibody. This will not work. The laser number 2 will detect the PE, fluorochrome, which give you the positive reaction. So it's very simple. You have beads coated with HLA molecules. When you have antibody, will bind to it. You use secondary antibody to detect the binding. Then the machine will tell you if it is positive and what is the specificity. And I will show you an example now. This is the exact example. I just pasted this diagram for simplicity. This is the readout of the Luminex machine. You have here different, this is for class 2, HLA class 2. You have DR and DQ and DP. And here you have the beads number, and coated with what? Like the first one here coated with DQ8. The one after that DQ9, DQ2, DQ7, DQ4. And here show you that this patient is positive for these particular beads or antigens. And here showing the MFI, which is the sort of titer. It's not real titer, but it gives you the reflection of the strength of the antibody present. It's very important not to mix it with titer. It is not titer. So again, bead number 7, it is here for DQ2, and here showing DQ2, bead number 7. This is bead number 7, showing specificity for DQ2, which is the one shown here. And the rest is exactly the same. It's very simple. You get a readout from the machine and it's telling you what is the specificity of the antibody present in the patient's serum. This is just to show you what I just mentioned earlier, that this is the antigen, single antigen beads presented on each bead. Again, MFI. And here, showing the threshold used to define strong, moderate, or weak HLA antibodies. This is very strong with very high MFI, close to 20,000. This is something less than that, if we can call it moderate or weak, and here we can call it negative. And very important to appreciate here that the threshold vary from one lab to another lab. What may be becoming or defined in one lab as it is moderate, maybe in another lab it is weak, or the opposite, it will be high. It depends on the lab calculated their own cutoff. Each lab should have his own cutoff for calling moderate or weak or very strong. When we using Dominic's technology, I am behind. When we're using Luminix technology, there's 3 categories. And the first one we use, which is HLA screening initial, we use set of beads, like 15 beads, for example, carrying HLA class 1 molecule, and another 15 or 20 beads carrying HLA class 2 molecules. And we run the test exactly, and the machine will tell you whether class 1 positive or class 2 or both positive. For example, if the machine tells you that it's class 1 positive, class 2 negative, you can go for next step of testing, which is using HLA phenotype or specificity, depend what you call it in your lab, in which each bead coated with haplotypes. And if the patient is not really highly sensitized, still you can tell the percentage PRA, like the old method, the specificity if there is no a lot of antibodies present with different specificities. And there is the test which we all like, HLA single antigen, in which each bead you use is coated only with a single HLA molecule, only one. For example, this particular bead coated with A2. And when you use single antigen beads, even if the patient is really very highly sensitized, the probability of telling what antibody the patient has is really very high. And it cannot tell you percentage, it will tell you only specificity. This is an example of antibody screening only. We have here our cutoff point. All these beads are below the cutoff point, so we call that this is a class 1 negative, and the cutoff point is 1.5. As you can see here, up to 115 is positive. So the patient is HLA class 2 positive. Everything in red is very friendly software is positive, here is negative. So this patient is sensitized for class 2 only. This is an example of a patient who is sensitized and he has a potential donor. The donor HLA typing is A1, A3, B57, B8, CW5, CW7, and we tested his serum whether he has antibody against the donor. So apparently when we did the antibody testing, he has more than one antibody, anti-B57, anti-A2, 69, B58, and 68. And as you can see here, he has antibody anti-B57, which the donor has. So in this case, we call this donor-specific antibody, and when we report it to the physician, we like to report the MFI, which is the mean fluorescent intensity, which is reflecting titer, but it's not a titer. This is another example in which there is sensitization. I mean, the patient is sensitized, and this is the antibodies he is sensitized to, and this is the donor HLA typing. But luckily, although this patient is sensitized, he does not carry donor-specific antibody. That doesn't mean he's better than someone non-sensitized, but much better than someone sensitized with donor-specific antibodies. Another example of Luminex readout. This patient here, he is sensitized. He has anti-B35, B53, B51, B50, and B57. And when we look at the donor, the potential donor for this patient, he is A3, B8, B35, CW5, CW7. So B35, and the patient has an antibody anti-B35-01 as a high resolution. So is this really donor-specific antibodies? If we stop here, we can say donor-specific antibody, but if more complex lab who is equipped to do high-resolution class 1, they can really tell whether it is donor-specific or not, which we, inshallah, can. So when we did the same donor for class 2 by high resolution, he turned to be that B801 and B3502, the allele, the Maha alleles talked about. So the use is now even for solid organ transplantation. So when we looked at the specificity of antibody, it is anti-B3501, but the donor is B3502. So in our lab, we can say this is not a donor-specific antibody. But if a lab is not really equipped with all the tools, it might be called donor-specific and might deny transplantation for this patient. The sensitization, it is a problem. 32% of the transplant list in the US, they are sensitized, and 14% of them highly sensitized, and it's very challenging. Challenging for the patient by extending his time on the waiting list, which means he will be staying a long time in dialysis. Also, there is a greater likelihood of death while waiting, because he is sensitized. Whenever there is a cadaver donor or even living donor, he has an antibody too that cannot be really solved with desensitization. Also, of course, reducing his likelihood of negative cross-match for the same reason. Even if this patient being desensitized, the graft survivor is really poorer compared to someone who is non-sensitized. And for laboratory means a lot of extra testing, which means also cost for the institution. This is just showing the median time for group of people. This is a study in UK. If you have a PRA less than 5%, your median time to be in the waiting list before being offered the kidney or transplanted actually is 788 days. As the percentage of PRA increases, the number of days will be also higher. So many people, unfortunately, who are very highly sensitized will die while waiting for the right kidney. This slide showing the deceased donor study, in which the same idea as previous slide. If they are non-sensitized, the 5 years graft survival is of course the best. If they are 10% or less, they are not as good as 0%, but they are still okay. But more than 10%, you start seeing a big difference. So as the number of PRA or the percentage of PRA increases, the graft survival will decrease as well. Back to the methods, I talked about CDC or CDC and AHG and Luminex. For CDC to be positive, you need to have enough or high level of donor-specific antibody to be positive. And for the method we are using also in our lab, even if it is with very low DSA titer present, or what we refer to as MFI, we will still be able to detect it, and we will inform the clinician that this patient is sensitized. Yes, not high titer, but you have to modify your regimen in treating him. The solid phase immunoassays, it has many strength point over the traditional method, which eliminates, of course, the need for lymphocyte and lymphocyte preparation. Very high sensitivity and specificity. It can be really automated and also unaffected by most non-HLA specific antibodies, because we use purified HLA molecules. again about automation. Reaction is on a continuous scale, so you can really tell not just with serology. You reported by 8 reaction strengths or 4 or 2. Here you have continuous scale, which help you to monitor also when you